Next presenter is Anna Taylor from UCLA, talking about microglia and dopamine function.
Great. Well, thank you so much for the invitation. This has been a great day. So my talk's actually going to kind of draw in on a lot of themes that we've seen already today. So I'm kind of excited for maybe some discussion. So as we've seen already, we know that drug-addicted state is associated with a hypodopaminergic tone, and Dr. Fowler presented some great imaging studies from D2 receptor binding. And not only does this hypodopaminergic tone drive the impaired reward behavior of the drugs of abuse, but it also undermines or drives the anhedonic state associated with drug withdrawal. And so we know that this sort of anhedonic or negative affective state is responsible, at least in part, for the high rate of relapse of in patients, drug-addicted patients trying to, in conditions of abstinence, trying to kick the drug. And so really in the study I'm going to present today is describing a novel mechanism that we identified that explains in the opiate-dependent model how the impaired dopamine system happens. And so we described this novel mechanism that incorporates activated microglia releasing BDNF, which impairs the inhibitory neurons in the ventral tegmental area. So that's what I gave you the punchline at the beginning. That's what I'm going to show you today. So first, just to really quickly review the fact that there is an impaired dopamine signaling and function in opioid-dependent states. This is an idea that's been in the literature for many decades. In the talk here, this is really one of the first studies that found this, and so in an animal model, they're basically just recording spontaneous activity of the dopamine neurons, and so this is in the bottom, the control animals, and in the top is the morphine withdrawn animals. And so you can see basically the top lines, there's much less spontaneous activity than the control animals. In the bottom sections here, we've also shown that there's a reduced drug-evoked dopamine release in opioid-dependent states. So on the left here, this is morphine, whereas if you give systemic injection of morphine to an animal already exposed to opioids, it fails to stimulate dopamine. And we've also shown this with cocaine. So again, animals that have been exposed to opioids, we give them a shot of cocaine, and we don't see this drug-evoked, this cocaine-stimulated dopamine release in these opioid-dependent animals. So this is really important because what this shows is that the impairment of the dopamine system in opioid-dependent animals is not because of a desensitization of the mu receptor. It's really a change in the activity or the function of the dopamine neuron in and of itself. And so the question is how? So there's been actually a really fascinating series of papers that have come out of Eric Nestler's group recently, where they've identified BDNF as a really important modulator of these dopamine neurons. So what they did here was, again, measuring the activity of these dopamine neurons in animals in the presence or absence of BDNF. So here, when you inject an animal with morphine, you get this really increased burst firing activity of the dopamine neurons, which then translates to a release of dopamine, and this drives reward behavior. But when you give morphine in the presence of BDNF, you no longer see this activation. So morphine can no longer activate the dopamine neurons. And so this really indicates that BDNF might be a critical modulator in dopamine activity. So the question then is, does BDNF contribute to this impaired dopamine activity in opioid-dependent states? And if so, where is the BDNF coming from? So that's where we started our study. So we took advantage of this fluorescent in situ hybridization with collaborators at USC out of Pat Levitt's lab. And basically, we took sections of the VTA and we labeled for BDNF as well as message, as well as a number of other neuronal markers in the VTA, including the dopamine neurons and GABAergic neurons. So what we found here was that in green is the BDNF, and then dopamine neurons, we used TH, tyrosine hydroxylase, and then GABAergic neurons are identified with GAD in blue. And so the green signal, the BDNF, we found while there was some co-localization with neurons, especially the dopamine neurons, there was a significant portion of this BDNF signal that was found in areas that did not co-localize with neurons. And you can see that with the yellow arrows. And so the question is, well, where is it? It's not just floating out into space. It's got to be contained somewhere. So we performed another experiment, and this time we looked at BDNF and co-localized it with an integrin receptor promoter. And so this is something that specifically identifies microglia. And so here we found that there was a significant portion of the BDNF message that co-localized with microglia. And not only that, when we quantified the BDNF signal and compared it to slices from naive animals and animals that had received chronic morphine, we found there was a significant elevation of the BDNF message. We also looked at protein, so we punched out the VTA from control in opioid-dependent animals, and we saw a significant rise of BDNF. And importantly, when we took opioid-dependent animals and pre-treated them with a microglial inhibitor, so in this case, minocycline, we could block this rise of BDNF. So together, these data really suggest that the opioid-stimulated BDNF is coming from microglia, and if we inhibit microglia, we can block this BDNF signal. So we then wanted to see, well, okay, is this microglial inhibitor going to be an effective way to restore dopamine activity? So I've just shown you the data that I showed you before. So again, this is in mice that we've treated twice daily with escalating doses of morphine, and then we test them 12 hours after their last shot of morphine. So they're in a state of withdrawal. And so in the dependent animals, if we give them systemic cocaine, we are not able to evoke this dopamine release. But when we look at opioid-dependent animals that have been pre-treated with this microglial inhibitor, we can restore the cocaine-evoked dopamine release. So we then wanted to look at behavior. So we used the conditioned place preference assay, which we've seen already, to probe whether cocaine reward is different in the opioid-dependent animals. And so this is where they receive repeating injections of either cocaine or saline on alternating days. And confined to one side or the other, and then on the final day, they are given free access to both sides of the chambers, and we measure the time they spend on the drug-paired side. So these are heat spots that are just mapping where the animal is spending their time on the post-conditioning day. So on the first day before they get conditioning, there's no preference. They'll spend equal amount of time on either side, but in naive animals, once they've been trained with the cocaine, they spend significantly more time on the place where they got the cocaine. However, we don't see this reward in the opioid-dependent animals, so there's no preference. They don't lend a preference to the cocaine. So this really is in line with the microdialysis results I showed you. Cocaine is not stimulating dopamine in opioid-dependent animals. Cocaine is not rewarding. But then we wanted to see, okay, does a microglial inhibitor restore this function? And in this case, we use two microglial inhibitors, minocycline, which is kind of your classical tetracycline, a really dirty drug, but it's what's most commonly used in the literature. We also use this novel microglial inhibitor, naloxone. This is an enantiomer of the opioid antagonist, naloxone, that doesn't have any activity at the opioid receptor, but it's a really potent and selective inhibitor of microglia. And in both cases, when we treat opioid-dependent animals with the microglial inhibitor, we can restore cocaine reward. So I'm making the argument here, or I'm trying to anyways, that microglia in the VTA are driving this dopamine impairment. But so far, all our microglial inhibitors have been systemic, so we wanted to really drive home the fact that it's in the VTA. So we injected a microglial toxin, MAC1 saccharin, and we can inject that directly into the VTA. And so when we inject our control toxin, there's no effect. We see the same thing. Naive animals, there's a cocaine place preference. This is lost in the dependent animals. Whereas when we inject the toxin to kill off the microglia, we restore cocaine place preference in the opioid-dependent animals. So that's the microglia story, but we're arguing that the downstream effector is the BDNF molecule. So to really drive home that BDNF is impairing cocaine reward in opioid-dependent animals, We took naive animals and injected BDNF into the VTA and assayed cocaine place preference. And sure enough, BDNF blocks cocaine reward in these animals. Now this kind of flies in the face of how we understand BDNF, which is generally thought of as a pro-learning molecule, and that's generally through the hippocampal actions where it improves learning and memory. So what we did as sort of a control experiment is we took naive animals and injected them with a systemic BDNF agonist, basically a TrkB agonist. And here what we found is this promoted cocaine place preference. So this is more in line with what we kind of understand the dogmatic view of BDNF. So our conclusion that we draw from this is that BDNF has a very circuit-specific effect. So while in general the pro-learning effects clearly dominate BDNF in the brain, specifically in the VTA, it impairs reward learning. So the question is how? And so this is my last few slides as you've really teased out the mechanism of this. We know that BDNF has a really important role in mediating GABAergic inhibition. So this has been shown previously in the spinal cord. So activation of the GABA-A receptor, which is a chloride channel, absolutely relies on low intracellular chloride to drive its hyperpolarizing effect. BDNF has been shown, so part of this sort of cascade is the potassium chloride transporter, which actively pumps out the chloride to maintain this low intracellular chloride. BDNF has been shown to cause downregulation of the KCC2. So when you have a downregulation of the KCC2, there's an accumulation of intracellular chloride, and this impairs the ability of GABA to be inhibitory. So that now when GABA binds the GABA-A receptor, chloride no longer moves down the concentration gradient into the cell, but it actually, in the worst case, can move out, in which case GABA would be depolarizing. So we wanted to see, we know there's lots of GABA in the VTA, so we wanted to see whether there was an effect of KCC2 expression. So I labeled KCC2 in the VTA. Interestingly, the KCC2 is found not in dopamine neurons, but in non-dopamine neurons, which I'll show later are GABAergic neurons. And sure enough, there's a significant downregulation of KCC2 in the VTA of opioid-dependent animals. I also looked at the function of KCC2, and I used a novel imaging technique using MQAE, which is a chloride indicator, so it fluoresces until it comes in contact with chloride. So when we take slices of the VTA, apply MQAE, we can image how fast the chloride enters the cell based on the quenching of the fluorescence. And so what we found is that when we look at naive animals and we look at the movement of the quenching of this fluorescence, it's pretty quick, right? So that's indicating chloride moving through KCC2. Whereas if we look at slices taken from opioid-dependent animals, this movement is a lot slower, indicating, again, a loss of function of the KCC2. Importantly, when we treat the opioid-dependent slices with a TrkB antagonist, we can restore the movement of chloride through KCC2. So again, one last experiment to kind of cinch it. I'm claiming that KCC2 is really critical in these effects, so we wanted to show this behaviorally. We took naive animals, we injected them into the VTA with a KCC2 inhibitor, furosemide, and assayed cocaine place preference. And sure enough, blocking KCC2 function reiterated the opioid-dependent state and blocked cocaine place preference. So just to quickly summarize, what I've shown is that in the GABAergic neurons, there's an impairment of the GABA-mediated inhibition driven by this loss of KCC2 function. So what that means is that these cells are now, the GABAergic cells are now hyperexcitable. So we know that the GABA neurons are incredibly important in mediating the activity of the dopamine neurons. And so when the GABAergic neurons are hyperexcitable, that's translating to an increased inhibition on the dopamine neurons and the hypodopaminergic function. And so it's the end of the day. I'm sure I've lost you, or some of you anyways, but if there's one take-home message you can remember, what I've shown is that blocking the microglial activation is a really effective tool at restoring the function of the dopamine neurons. And so both minocycline and plus naloxone are available for use in humans as we speak. And so it's a really obvious tool to give to patients to sort of improve the negative affective state and to improve or decrease the rate of relapse of these patients. So thank you very much. I'd like to acknowledge my postdoc advisor for 7, my collaborators, and our funding. Thank you.
Great talk. Thank you. Very nice work. A lot of data. We'll take one question. What were the 2 drugs at the end you recommend?
Minocycline,
right.
which is kind of toxic, so that's not great, plus naloxone.
Okay. So monocycline is certainly not FDA approved for its indication, but naloxone, some controlled substance use.
Totally.
Great.
Yeah,
Great.
for sure.
Thank you so much.